Hello friends, welcome to my YouTube channel. Today I will discuss about overview of nucleic acid. So let's begin. In this session I will cover introduction of nucleic acid, nucleotide components, nitrogenous base, structure and their nomenclature, other nitrogenous base, sugar and binding of nucleotide. In my next video, I will discuss about the DNA and the double helix DNA and triple helix DNA and analogs of nucleic acid. So, at first you have to know what is nucleic acid. Nucleic acid is the polymer of nucleotides. So, we know the nucleic acid is two type. One is DNA, other is RNA. DNA means deoxyribonucleic acid and RNA means ribonucleic acid. So the DNA is the chemical basis of heredity and it can be regarded as the reserve bank of genetic information. The DNA is also responsible for the maintaining the identity for different species over millions of years. But the DNA is organized into gene that controls the protein synthesis by mediation of RNA. You know the following process that the DNA to RNA, RNA to protein, but these three biomolecules mix. These three biomolecules constitute the central dogma of life. But here it is very important to know about nucleotide because the nucleotide it is the one of the structural component of nucleic acid. So let's discuss about the component of nucleotides. Give me some time. This is a component of nucleotide. Nucleotide, here are three components. One is nitrogenous base, sugar and phosphate. Here you can see there are three components: nitrogenous base, sugar and phosphate. Here the nitrogenous base, there are various type of nitrogenous base. By majority we know the nitrogenous base, purine, pyrimidine, but there are different kinds of bases are also found. uh, Later I will discuss about them. And the sugar, here the sugar is also pentose, and the phosphate, here you can see the phosphate is PO4, PO4 2 minus. So here we also contain two negative charge. So the DNA also acidic for this phosphate group and also carry the negative charge for the presence of phosphate. So it was the component or basic component of nucleotide. And let's learn about nitrogen base, then sugar, and then phosphate one by one. So let's see. The nitrogenous base, the major part of nitrogenous base is purine and pyrimidine. Purine also contain adenine, guanine, but pyrimidine also contain cytosine, thymine, and uracil. Let's see their structure. Before see their structure or each and every one of them, but let's see their general structure first. Here you can see this is the general structure at the A. This is the general structure of purine bases. In the case of B, you can see this is the general structure of pyrimidine bases. Let's use the laser pointer. Sorry, this is the laser pointer. Just wait. You can see this is the structure of purine bases and this is the pyrimidine bases structure. But here you can see the number system. The number system in this case of the purine, you can see this is the anti-clockwise 1, 2, 3, 4, 5, 6, and here you can see the number system is clockwise 1, 2, 3, 4, 5, 6. In the case of purine, you can see this is the this structure, the two also contain the heterocyclic ring, but here you can see this also this also anti-clockwise direction, numbered as anti-clockwise direction, but the pyrimidine is also numbered as clockwise. So here it is very here is this difference from the structure, and this rule is also accepted international level. So let's discuss about the structure of uh, here there is another term, it's called tautomerism. What is tautomerism? Tautomerism means the existence of a molecule in an lactam form and lactim form. Lactam form also known as keto form, and the lactim form also known as enol form. Here you can see a oxy group is present here. A oxy group is present here, and the hydrogen group is also attached with oxygen and produce a lactim form here and makes a double bond between N and C. Because this bond will break, and this there will be they produce a bond, so it makes a double bond. This is a lactin form. It's a lactin form and a lactam form to lactin form, and this lactam form. This form also present for adenine. Sorry, not except adenine. They also show this type of tautomerism. You can let's see their structure. You can see this there are this type of oxo group is present at the guanine, and here is an H group is present. They can show the tautomerism. In the case of cytosine, they also show tautomerism because there is a oxo group is present. Where you can see the oxy group is present or oxo group is present, then there will be such a probability of showing this tautomerism. Like let's see, you see this is adenine, guanine, cytosine, thymine. Except adenine, the guanine, cytosine, and thymine uracil, they also show for tautomerism. But here you can see the at the physiological pH, they all show here the lactam form. That the what is the meaning of physiological pH? Physiological pH means the pH at 7.4. It is called the physiological pH. Here you can see this is the adenine, and adenine there you can see this the NH2 group is at the sixth position, so it is called 6-aminopurine. Here you can see the alkylase guanine. You can see, so the general structure for the adenine and guanine is remain same. It will remain same, but here you can see this is the NH2 group attached at the sixth position, so this 6-aminopurine. Here you can see the 2 amino and 6 oxypurine because the if I number here this 1 2 is so the 2 position is amino, and at the sixth position is oxy group is present. So oxy group is present, so it's called 2 amino 6 oxypurine. And here you can see this is the cytosine. In the case of cytosine, this number system is also different. And you can see this is the number system also clockwise at a clockwise direction. So number we will give the number it 1 2 3 4 5 6 1 2 3 4. So at the if I give it number, let's see this picture of the numbering. Here you can see this is 1 2 3 4 5 6. So number system is here quite different, and here you can see this number is also anti-clockwise. So here you can see this is the cytosine. In the case of cytosine, the 2-oxy 4-amino pyrimidine. So it is the structure of pyrimidine. So it is called 2-oxy. Here you can see at the point 2 oxy is present at the 2 position, and the 4 amino is present at the 4 position. So 2-oxy 4-amino pyrimidine. In the case of thymine, it is also it is 2,4-dioxy, because here 2 and 4 position 2 oxy group is present, so 2,4-dioxy 5-methyl pyrimidine is present, and there is a methyl group is present. But it is very interesting to know that if I cut down this methyl group from here, you can get uracil structure. All are same, but here you can, if you cut this CH3 or methyl group from here, you can get uracil. So it was the general structure for the general structure of major nitrogenous base. Let's discuss about minor bases. Minor bases are found in nucleic acid beside their bases, and the minor bases are about they're like 5-methyl cytosine, N4 acetyl cytosine, and N6 methyl adenine, N6 dimethyl adenine. These are and uracil. These are also known as the minor bases, and we do not know the proper function, but it is believed that that it is believed that this unusual basis in nucleic acid will help in the recognition of the specific enzyme. So it also helps the recruitment or the recognition of the specific enzyme. It is believed that. Uh, so next, the base other base there are also present other bases. These other biological important bases are hypoxanthine, xanthine, uric acid. Let's see their structure. Here we can see this is the hypoxanthine, xanthine. The hypoxanthine and xanthine, if you look at its structure, they also uh, they get a structure is purine structure. This here the general structure of purine. You can see this is the structure of purine bases. Hypoxanthine and xanthine, they are intermediate of producing, while producing of purine bases, they also act as the intermediate. So it is at the position of 6 oxyos present, so it is 6 oxypurine. And you can see the 2 and 6 positions, so it is dioxypurine. And you can see here and here and here there are 3 oxys present, so it is 2,6,8 trioxypurine. But here if you notice that these two are former of it is also form a sorry, it is it acts as an intermediate while the synthesis of purine. But uric acid, that is the final product of purine degradation. So uric acid is the final product or end product of purine degradation. Let's see about sugar. You know that sugar in this case also present at pentose. Sugar is also present here. It's called furanose. So here you can see this is the sugar for ribose and deoxyribose. But if you notice this structure, the all are the same. All the structure is same. But here just a different is here at the C2 position. You can see there is a OH group is present here. But here you can see the lack of O is present. So there is no oxygen group is present. Here you can see there is an oxygen group is attached with hydrogen. So it make a OH group, but is no wedge group is also present. Only hydrogen group is present. But if you draw here a line or a plane, if you draw here, uh, when the OH group is upper from the plane, then it will be called beta anomer. It will show beta anomer. But if the OH is lower from the plane, then it will show alpha anomer. So this structure also for ribose, and it is deoxyribose. But here remember here um, we also this number this system not only 1 2 3 4 5. We not use this 1 2 3 only this number. We also use prime. We also use prime, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, because just we want to make a difference from the nitrogenous base, because they also contain this carbon, and the carbon there are also present some carbon and in their heterocyclic ring. For to make a difference from them, we use prime the sugar structure. Here we see the binding of nucleotide. It is our last topic here in this video, and my next video I will discuss about double helical structure of DNA, and other is the analogs of nucleic acid. Here you can see this is the adenine structure, and here if you see this is the adenine, and this is attached with this sugar. This is called ribose sugar. This ribose sugar is also attached and at the 1 prime. So adenine attached at its 9 part will attached with the ribose 1 prime part. So it is the when adenine attached to ribose, it is called adenosine, and the phosphate is attached at the 5 prime, so it is called adenosine 5 prime monophosphate. And here if you see this structure at the thymine, the N1 part is attached with the first prime 1 prime part of ribose. So this is also the thymidine. It's also called here you can see that if there is a thymidine phosphate, there is thymidine, and another phosphate is joined as usual of the 5 prime part. So here we can see this is it is another phosphate is attached, so it is called thymidine 5 prime monophosphate. But here if you see this structure, and if I add here another 2 phosphate here, then it will be called adenosine triphosphate. adenosine. So here is only adenosine. Adding of the second and third phosphate give adenosine diphosphate and adenosine triphosphate. So just we know that it is if I add is another phosphate, second another phosphate, that will be called ADP, adenosine diphosphate, and if I add another two phosphate, then it will be called ATP because of presence of three phosphate. The 5' position it is called ATP, adenosine triphosphate. So it was our last topic. Let's read again. Binding of nucleotides. You can read and note down. The pentoses are bound to the nitrogenous bases by the beta in glycosidic bond. Here, just the N9 position, that is of the presence in the purine ring, and the C1, the first prime position, 1' position, that is attached N9. That is also present. There is also bonded with the N acid N glycosidic bond. They also bound by N glycosidic bonds, and the in the case of here the pyrimidine of nucleotides, the glycosidic linkage in the between of N1 and the pyrimidine and the prime here, the C1 prime of a pentose. Let's see. Here you can see the N9 and C, the ribose 1 prime. They also attached by beta in glycosidic bond. But here if you see, there is another glycosidic linkage is present in the case of first prime ribose and N1. So it was it is we have completed our session. So thanks for watching. Goodbye.